introduction we discuss. So those organism or those substance which are micro, means they having the size in micro scale, they are under this system of the study. First thing which we study, that which of the substance are smallest in our world, that are prions. We consider as a prions as a smallest smallest particle. Prions are not cell, they are what? They are the some proteins. Here we discuss by taking this is a neuron, brain neuron, which having the nucleus synthesize DNA. From DNA the proteins are comes out, and the protein, the primary structure of a protein we know alpha helix. So when alpha helix and as well as beta plated are the primary structure of the protein. So when alpha helix and beta plated both are primary structure, but they having some configurational difference. When the alpha helix have one important thing is, remember that protease, the enzyme which breaks down the alpha helix, not beta plated. That's why the beta plated chain of the protein are abnormal chain, and which called as prions. Why it is called as prions, we discuss. So what happened, that why beta plated are known as abnormal protein? Because the protein, there was a concept was given by the scientists that in our nucleus, from nucleus the protein which are synthesis always degraded by the protease and new protein are synthesized. Okay, in that way we called as a protein turnover. So we discussed that what is protein turnover. Protein turnover is definition is that when a DNA synthesizes the protein, these are undergo degradation by the proteins and a new protein are simultaneously synthesized occur. But the beta plated protein are not, we discussed that beta plated protein, these protein are not break down by the protease. Why? Because have some difficulty, because they are abnormally rearranged chain are present which are not break down by the protease. That's why they are not degraded, and when they are not degraded, they are remain in our tissue or in our cell or in our body in a store form, because they are not degraded. That's why what happened, this becomes a abnormal and deposited in our tissue. So a case which we found that amyloid body, means beside prion, whatever other proteins are beta plated, they are amyloid, which we discuss in our pathology. That what is amyloid? Amyloid are any protein which are beta plated present extracellularly and deposited in the tissue. Because they are resistant to the proteolytic enzyme, that's why they are deposited in the tissue. And the prions, we should remember that they having no DNA or RNA, they are simple proteins. But if the protein are abnormal DNAs, they become beta plated, and these beta plated mixed with this alpha helix and became a chain which are stored in our body. So here you can see the beta plated alpha chain, they mix and they undergo abnormally forms the prion related protein, means they corrupt the alpha helix and becomes corrupted protein are synthesized. That's why they are abnormal protein, and abnormal protein when mixed with the alpha helix normal protein and becomes abnormal. In that way they are increase their size and increase their number. Okay, so we discuss what is prion. Prion are nothing but the infectious agent which are misfolded beta plated protein. And beside prion beta plated, we discuss also a amyloid which is also a beta plated, and it causes the damaging our tissue by producing a process which is known as amyloidosis. Okay, now from prions we discuss what is prion, what are the importance, and how it is a transmissible. It is, what are the properties of a prion? They are transmissible, they are spongiform, and they causes transencephalopathy, some disease they also causes. So in this way we discuss the smallest particle, that is prion. After that we go towards another one. They are bacteriophage, plasmid, transposon. They are also the small microbiological particle. So first thing which we discuss plasmid. After that we can undergo bacteriophage and transposon. So what is plasmid? So here bacteria capsule, then it is the bacteria chromosome. Okay, this is the bacterial chromosome. Beside the bacterial chromosome, another a double-stranded ring structure was found, which is known as the plasmid, and it has a virulent property. If the plasmid contain a virulent gene, we called as those plasmid as a virulent plasmid. Okay, we discuss what is plasmid. Plasmid are extrachromosomal double-stranded DNA ring structure which having the capable of itself own replication. They do not depend upon the bacterial chromosome for its replication. They itself goes replication. And what happen if the if we state that this is a bacterial capsule and it have a bacterial chromosome, inside the bacterial chromosome if a extra chromosomal DNA which is present, which is known as a plasmid. What happen? The plasmid is it having own DNA, so it can produce protein, and those protein if got mixed with the bacterial chromosome, they also causes virulent bacterial chromosome. Okay, so in that way the plasmid how infection occur, it can combine with the bacterial chromosome and become the virulent property of the bacterial chromosome, as well as when bacteria undergo the binary fission, it can transport its protein towards bacteria and causes the virulent nature of the that bacteria. Okay, in that way the plasmid causes the virulence in one after another. So what is the role of a plasmid in relation to drug? So how plasmid causes drug role? So it can, due to its having the virulent property, the protein which he secreted, it can acts with that if we take this is a penicillin, penicillin having beta galactide ring. When there will be the protein can bind with that beta galactide ring and causes destruction. In that way the plasmid causes antibiotic destruction and causes the resistance towards the antibiotics. Means bacteria which are infected to our body, if they are having the plasmid which are virulent type, then these bacteria are not destroyed by the antibiotics, because the plasmid causes the destruction of that antibiotic drugs. Okay, we can discuss the plasmid in relation to drug. So how it causes infection in our body? Plasmid are the infectious agent which we discussed before, because the plasmid enter our bacteria. Here you can see the enter into our bacteria and causes the adhesion of the bacteria, as well as they produce the virulence, and which enter into another replicated bacteria and causes indirect disease. So this causes indirect disease, not directly it causes any type of disease, but indirectly it causes disease. That's why we know how the plasmid, what is the plasmid, what is the relation of plasmid inside the drug, how plasmid acts in our body. Then we go towards the bacteriophage. What is bacteriophage? We discuss by giving an example of a Corynebacteria diphtheria. So Corynebacteria diphtheria is a healthy bacteria which presents some people inside the throat. What happened when a Corynebacteria diphtheria bacteria having the bacteriophage, when they infect in that bacteria, what it causes? The bacteria causes release its toxin by pouring the bacterial cell and causing the toxic genes produced, and these toxic genes are comes out from the cell, and extracellularly it causes exotoxin protein production, and these exotoxin production causes diphtheria disease. So is the substance which bring the genetic material of infectious and enter to body and causes indirect infection. So in this way we discuss the bacteriophage and the plasmid causes no direct role in infection, they indirectly causes infection. Then we discuss the transposon. What is transposon? Transposon, its name itself says that they are transporter substance. So in a bacterial chromosome, if we see that any type of a gene coded with having a leg, means movable substance if coded, then this is known as a transposon. So what is transposon? Transposon is that substance which having the role DNA fragment which having the jump. It can jump towards the bacterial chromosome or any plasmid. It can enter to the plasmid and coded with plasmid. In that way they are known as a mobile jumping gene. So we discuss what is the term regarding of the brief introduction of these terms. these are the smallest, then it is intermediate. If we think, suppose it is smallest, then it is intermediate, and it is somewhat larger than this substance. So virus is nothing but it is not a organism. So remember that virus is a particle which having DNA or RNA or protein, and some virus are covered with lipid. Why it is not an organism? Because organism are those which having the cell. Virus do not have a cell, because besides the host, virus is inert. That's why they are the particle having DNA or RNA covered with a lipid capsule, that is known as virus. After that, we discuss the bacteria, fungi, ectoparasite, helminthes. These are eukaryotic infectious organisms. So all bacteria are not infectious. Some bacteria who are coded with the infectious substance, they are infectious towards our body. Then fungi, ectoparasite, helminthes, these are the substance. These are eukaryotic substance cell, and this is eukaryotic cell. The prokaryotic cell having the chromosome in a double standard rounded, but eukaryotic cell, they are nucleus are present which is rounded. The chromosome which are present here the circular, here the linear. So here we can discuss by going in that way. The nucleus are present in the eukaryote, but prokaryote do not have any nucleus. So this is eukaryote. The envelope, there is some nuclear envelope is present in the eukaryote, but prokaryote do not have an envelope. Double stranded DNA is present. Here you can see the eukaryote having double stranded DNA. Here no double stranded DNA is present. Then linear chromosome are present in the eukaryote, but in the prokaryote the chromosome is circular. Then it is having the histone. Here you can see the chromosome containing the histone protein, but in the prokaryote there will be no histone, and there is a multi-chromosome is present in one nucleus. Here is no chromosome is present, single chromosome is present. Then DNA with histone, here DNA is not present, so there will be no histone. Then nucleus is present here, here no nucleus is present. Then here the ribosome is also present, which is 80S type, it is 70S type ribosome. Okay, these are the difference between eukaryotic and prokaryotic substance. Now, after discussing these prions, bacteriophages like structure and the virus, now some and also the prokaryote and the eukaryotic difference. Now we go towards the bacteria classification. So inside our microbiology, bacteria are classified into two types. One is gram positive cocci, means all bacteria are all positive bacteria are cocci, means all positive gram positive bacteria are cocci except meningococci, genococci, veio and moraxella. These are gram negative cocci. Okay, then gram negative bacilli means all bacilli are gram negative. Okay, gram negative except Mycobacterium, Anthracis, NACD, these are the mnemonic of gram positive, that is gram positive bacilli. These are the exception. Okay, then we go towards the fungus. Fungus are classified according to their cell morphology into four types. One is yeast, means true yeast, then yeast like fungus, then molds and dimorphic. These are the fungus classification. Then protozoa classification is occur according to their size, that is amoeba. They are mainly protozoa which comes under the amoeba are two types, free living amoeba or intestinal amoeba protozoa. Then ciliates, they having no something like difference was not found. Sporozoa, protozoa and flagellates, they may be present. Flagellates also classified into intestinal flagellate, vaginal place flagellate and blood and tissue flagellate. So in this way we discuss only the basic classification of this substance. And now what are the example of true yeast like Cryptococcus, yeast like Candida, molds are Aspergillus, dimorphic are Penicillosus. These are one example of different kinds. Then in parasite or protozoa case, the amoeba, free living amoeba, acanthamoeba, intestinal amoeba, entamoeba histolytica. In ciliates, that is Balantidium coli. In sporozoa, the blood sporozoa is Plasmodium, the tissue sporozoa is Toxoplasm, the intestinal sporozoa is Lycospora. Then in the flagellate case, the intestinal flagellate are Giardia lycopodium. Then vaginal is Trichomonas. In the blood and tissue, Leishmania. These are the all one example of a different kind of a protozoa and a fungus.